So, okay, so we're doing transport in mammals. Let's start off with the learning objectives that we have. So the learning objectives essentially given in your syllabus include the general system, the arteries, capillaries, veins, oxygen curves or oxygen, how is it carried, how everything happens, and then the anatomy of the heart, how the heart performs its role, how the heart itself is used, etc. So let's see. Okay, so the first thing you need to understand from this topic is that you need to be able to appreciate that we have the circulation in our body called a closed double circuit, which means now what does these are two words or actually three words. Circuit means it's complete, there is no gap in it. But what does the word closed and double mean? Closed, again, the same thing, that it's totally closed. Double means that there are two parallel circuits taking place, and in physics, being parallel is important because what happens in parallel circuits is that the resistance is highly reduced, but the power being provided to each of the circuit will be very high and equal, right? No matter what happens. So the two circuits that are important to remember are the systemic and the pulmonary circuit. Now it's a time of COVID, so you know what pulmonologists are. They're basically doctors of the lungs, which is why lungs are basically is basically generally known as the pulmonary system, right? So when the blood passes through the lungs, it's called a pulmonary circuit. It's a circuit because it comes from the heart and ends in the heart. Similarly, the systems of the body are known as systemic, and as blood comes from the heart, returns to the heart, goes in every system of the body, it's called as the systemic circuit. Now the pulmonary circuit starts, and the heart is divided into its four chambers. There's a right atrium, a left atrium, a right ventricle, and a left ventricle. So let me tell you one thing very clearly here. Whenever you are looking at someone or at an image, this side is always going to be the right side, and this side is always going to be the left side. So this thing should be instilled in your mind. There should be no confusion about it. This side is always the right side and this is the left side. So if you divide the heart, this is the right side, this is the left side. The right side is always deoxygenated and blue. It is going into the lungs to get oxygenated, from which it comes back after being oxygenated and is given into the body. Now, what are the two circuits as a whole? The two circuits as a whole are very simplistic. Essentially, all happens in the pulmonary circuit. Deoxygenated blood enters lungs, gets oxygenated, returns to the heart or towards the left atrium. And again, remember, entering tract is always an atrium and the leaving tract is always a ventricle. The second circuit, again, very easy. Blood that was entering into the oxygenated part of the left side leaves the left side to reach the body in a systemic circuit. And the circuit is completed when this blood comes back towards the atrium. Like I said, receiving circuits are always atrium. Okay, so let's see what we have further on this topic. Now this topic, if you understand that the circuit is a double closed circuit, two circuits occurring together that are closed on themselves, occurring parallelly, then you should need to understand the individual components that maintain the circuit. Now the individual components, let's start from here, are essentially the artery, vein, arteriole, right? I do not need to go into details of these, but I will tell you the important parts of these. So the first important part you need to know is that arteries carry blood away from the heart. So whenever you have blood being carried away from the ventricle, whether it's this one or it's this one, it's going to be called an artery. Whenever it's coming back, it's going to be called vein. That is one thing you need to know. Even in the fetus or in the baby that is growing in a woman's body, what happens is you look at it from the perspective of the heart of the woman. So whenever there's a vessel coming back towards the heart, it will be called, what will it be called? It will be called a vein. And if it's a vessel going away from the heart, it will be called an artery. Is this clear? Is this concept clear? Please don't ever lose this concept from your mind, okay? So that is one essential component. Now you need to understand how the system works. The system is very easy. You have a bunch of arteries that become, so what essentially happens is, let's draw it over here. You have one artery which divides into a bunch of smaller arteries called arterioles. These develop into a very dense network of capillaries. which basically perfuse the organ. This portion here, the capillaries, is what perfuses the organs. When the deoxygenated blood joins together into vessels, these small vessels that are joining together are essentially called venules because they join together to form a vein. And this vein will go to the heart, from which it will go back into an artery, and this essentially will then be arteriole, capillary, venule, and veins. So these basically baby arteries are arterioles, baby veins are venules, as simple as that. Okay. Now you need to know the difference in them. So essentially, it's very easy to recognize the three of them apart. Arteries have a very thick wall and a circular lumen. Now, first thing to understand, a very thick wall, because the wall is made up of a lot of elastic and smooth muscle fibers, because it has to see, it is a vessel going away from the heart, like it's illustrated over here. It has high pressure blood, which means that it needs to withstand that pressure without being tore apart, right? So it needs a large number of fibers to support it. So it has a very thick wall. And when something has a lot of stuff in it, if it's very thick, then its lumen is obviously very small. And when it has a lot of stuff in it that is not flexible, that is hard, and makes sure that even if high pressure of blood exerts itself, it maintains the shape. Let me tell you one more thing over here, a concept that you need to understand now and right now forever. Arteries tolerate high pressure of blood. Veins tolerate high volume of blood. To withstand high pressure, you need a circular, regularly shaped lumen and a thick wall. To withstand high volume, you need a very irregular lumen that can stretch and allow volume. You need it to be flexible, disorderly in shape, and you need a very thin wall so that lots of volume can be accommodated in it. And that adds up in the description. Now you need to understand the description of how any wall is made up. An artery wall is given over here as a description. Follow my pointer, please. So it's made up of very simplistic stuff. There is a basement membrane. Basement membranes essentially are single cell membranes that make sure no toxic substances or other substances enter they are present in organs, in arteries, in everything, right? They are basically they're like a screening membrane that make sure it's made up of cells and large number of collagen type 4. You don't need to remember these. I'm telling this to you for your information, right? You need to know this because this is one of those topics that is very heavily related to medicinal concepts as compared to the cells and the details of those enzyme, etc. So you need to know these things by concept rather than by rote learning, right? So you need to understand that any structure in contact with an external surface, like I told you, if we have a structure over here, let's draw this and try to understand this clearly. So if we have a structure over here, we have a structure like this. Now this structure is going to have something that's going to protect it from the stuff that is inside and something that is going to protect this stuff from outside. This structure, the inner protection, they might be single layers or multiple layers. The inner protections are called endo. So the endodermis, the endo, everything endo is the innermost. And the outermost, like I told you, ecto or exo or something like that. Okay, you'll get to learn these words very easily over time. Similarly, within these organs, within the endo, part of the endo or within the endo is something called a basement membrane that is given right here. This basement membrane is essentially collagen and cells. And what it does is it's basically a protective membrane. It's the basic structure. Now, the components of an artery wall is basically this basement single cell layer membrane followed by three layers. The innermost layer or the intimate layer called the intima, a layer in the middle or medium layer called the media, and an external layer called an externa. So an tunica and tunic covering basically. So tunica intima is the innermost layer, tunica media, the medium layer, and the tunica externa, the external layer. All you need to know if you understand this is that how these correlate with this image in your book, and that is it. So how does this correlate? Let's see. He says that an artery has an inner layer, which is endothelium. Like I told you, this endothelium is basically the basement membrane. Endo, the innermost layer of single cells, right? And endotheliums are always squamous epithelium. Squamous epithelium means that they are not regular. For instance, some cells you see are, if you've looked at images, some cells when you look at them are these square shaped, right? These are called columnar epithelium. Some cells are flat. These are called urothelial, like in the ureter, etc. But some cells have random shapes. Their shapes are adjustable and they adjust according to blood flow, according to organs. So this single layer is known as a squamous layer. And when this layer is on the inside of a capillary, if this is the inside, then this must be the intima, this must be the media, and this must be the externa. Intima, media, and externa, right? So if this, if to Okay. So for this reason, this part, this one layer is known as the basement endothelium of squamous cells, and it's smooth single layer of squamous cells or squamous epithelium. And like I told you, epithelium again means the same thing. Epithelium, endothelium, it's the internal most layer. Exothelium, ectothelium, these are the external most layers. So just a word play, let's say, which science loves to use just so normal people don't understand this. Okay, so you can imagine it that way, just to make sure. The other day we were talking about, we were discussing something in clinic and somebody asked us, okay, why do we keep referring people who have blood pressure as hypertensive and not just simply have people who have blood pressure? And so our overall conclusion was that we don't want other people to understand us. So jokes apart, yeah, so we have words that specify what is what. Then we have a middle layer, which is made up of elastic fibers, smooth muscle fibers, and collagen fibers. Now, let me tell collagen is a maintenance substance. The important thing here is the elastic versus smooth muscle. Some arteries may have more smooth muscle, others may have more elastic fibers. What difference will that make? Let me tell you the difference here. Elastic means that it is just there to stand that high pressure and rebound. Smooth muscle, however, means that it is a muscle, so it can contract to reduce the size of the lumen or dilate to increase the size of the lumen. So arteries with more smooth muscle or high in smooth muscle will have different properties. They will control blood flow amount, whereas elastic ones have generic properties. For instance, big arteries should be elastic. Why? Because their job should be to sustain high amount of pressure rather than change their shapes to provide that blood to other organs. So I hope this point is also clear. If it is not, I can explain it again. And then the final most layer. So I'm giving a very quick lecture this time because all of you have done this topic before. I want to make sure that I cover the tougher aspects as compared to the easier ones. Okay, and secondly, make sure you, whatever I tell you, make sure you write it down in your notes, because the information I'm giving you is more important than the stuff on the slide, because this is right out of your book. And the external layer is again collagen fibers. I told you, maintenance fibers, elastic fibers to make sure it doesn't, burst open, and then other substances, other fibers just to support it. External layers always give mechanical rubbing support. They give support against pressure, against tearing, against infection. So that's basically it. So the most important part is the medial layer, which has all of this structure, and the inner layer has one important structure, which is it consists of the basement membrane internally, and this basement membrane is squamous epithelium. Then similarly, we have inner layer, just like the artery. What is that supposed to mean? It means it's just squamous epithelium that we term as the endothelium when we talk about these three vasculature. The lumen is large, the middle layer is thin, has smooth muscles more than elastic fibers, just to be able to control the diameter, but some elastic fibers are present. And then the outer layer is mostly collagen fibers, no need for extra elastic and etc. We need to make it as thin as possible. The last one is the capillary. Capillary's job is just to provide everything that we have to the organs, which is why it is literally one cell thick. If you can see, it's made up of 3 cells, to form into a circular capillary. And each, what does one cell thick mean? It means if my hemoglobin is this red dot, and there is an organ over here on the outside, it just needs to cross the cell membrane of this one cell, two cell membrane, to go in and to go out to reach this organ, which means there aren't a layer of cells here, there's layers of cells, fibers, etc. So is this slide clear? This is the most important slide in this entire topic right now.
Yes, teacher.
Okay. Abdullah, is it clear for you as well?
Right now Abdullah isn't here, so he's kind of busy.
Okay, that's fine. That's fine. Okay. So then this is further descriptions. These descriptions, I hope, will help you understand this better as well. Look at the capillaries, appreciate the thick layers of the arteries, the thinner layers over here. Look at this capillary. Can you see one cell thick, and then the lumen that has no integrity, very less, each layer is thin, each layer is thick, lumen has a lot of integrity. Again, this is to describe the just like I told you, arteries give smaller arterioles that give capillaries that are one cell thick, capillaries joined together to form venules that are baby veins, which join together to form veins. Okay. Now, this is a very important slide. This should be important because you need to be able to recognize these three structures. So which one is an artery? Is this an artery or is this an artery?
The one on the right is the artery.
Okay, yes, that's correct. This one is an artery. Why is this an artery? Again, circular lumen, thick, not disproportionate, maintains a thick wall as compared to this one. Now, I can also see capillaries here. Can you try to find a capillary for me? Can you see a capillary?
The left one is the vein or capillary?
This one is a vein. Capillaries, remember, one cell thick, so very small. Do you appreciate this small structure over here?
Oh, yeah, over there. Yeah,
see, look at this one, very small. These smaller ones are essentially capillaries. Look at the one cell thick diameter, very small, perfusing the organ, okay? Again, for arteries, we can appreciate many cells over here. These dark dots are basically nuclei of the cell. This is vein. Now this is a vein. Look at the lumen. It has been folded over here, such a thin wall. And look at the inside. The inside looks like it has some fluid in here. The dots on the inside show that there are some cells floating in on the inside as well. Like I told you, the dark color dots are basically nuclei of all the cell. Can you appreciate how close together these cells are, how proportionate, how thick the wall is? If this is the intima, this is the media, then this is the externa, right? This one has very small wall, and this capillary is not drawn to size with these. Why? Because capillaries are very small. And can you appreciate the cell? Look at this one capillary. It has, this is the cell and this is the nucleus of the cell. Can you see how tiny it is? Just made up of three cells. I can see three nuclei. And on the inside, the dark shading areas are probably cells stuck like next to the capillary wall that got stuck while sampling. And these can have just to cross this distance to give oxygen and nutrients to the organ. So very easy stuff, right? So again, this is how your book describes your middle layers and all the layers. It's very easy to go through them if you understand these definitions in detail. It's very easy to understand. Squamous epithelium, like I told you, it is flat cells that form the lining on the inside of these hollow structures. Smooth muscles. So I hope you know the difference between the type of muscles. I think I have a slide on it as well. Let's see if it comes up. There are three types of muscles essentially. There is muscles that you can control, that you can change the activity of. Those are your striated muscles, or the muscles of your arms and legs. Then there are smooth muscles that you cannot change the activity of. They are not under your control. These muscles are in the, let's say, the gut, because you cannot, make the stomach go faster, or the intestines go faster, right? There are those muscles. And then finally there are muscles of the heart, that are cardiac muscles. Okay, so just three types of muscles that you need to know. Again, we talked about elastic arteries and muscular arteries. So elastic arteries have a lot of elastic, they stand high pressure. Muscular arteries, you can see, can change their lumens very easily. And arterioles are so much smaller than arteries, but they still have so much muscle fibers surrounding them, as you can see, as compared to this capillary that has nothing around it. So again, very easy concept, nothing difficult if you understand how elastins and muscles work differently. Smooth muscles control the diameter, elastin just there to withstand pressure. And this is another image just to understand this concept well, and you can appreciate the smaller artery here as well. Okay, then finally we need to understand these electron diagrams. In topic 1 we talked about diagrams that showed us through electron microscope that are not colored. So you can see it shows clearly the demarking of the inner surface of single cells of the endothelial. The elastic fibers can be seen here in the middle layer, smooth muscle and elastic fibers. And then finally, we have an external layer, and the RBCs can be seen here as well. So there are two terms you need to understand with muscular arteries: vasoconstriction or dilation. I've already told you what it is. Vasoconstriction, vaso means vessel. Constriction means that the muscle will contract and the lumen will become smaller, right? Because it's contracting. Vasodilation means the muscle is going to relax and the lumen is going to become longer, more blood can come into it. Okay. So I have a question. Suggest why there are no capillaries in the cornea of the eye. So, and I tell you that there are no capillaries. You need to tell me why. And the second thing is, how do you think then the cornea is supplied? So, any guesses?
I think it's filled with like water.
Yes, that's a
The
correct
cornea, right?
answer. Yeah.
Yeah.
So the cornea is basically the fluid in our eye is very rich in oxygen and nutrients. And that's very important because that's how it supplies just by diffusion. But why are there no capillaries in the cornea?
Because water and oxygen is like the larger molecules, it can be in capillaries.
Okay, no. So capillaries are a faster way for diffusion actually, which is why rest of the body uses that, right? The water dissolving is not always very a safe option, let's say, because you don't know, you can't reach every corner in that. Some corners may be missed, can lead to necrosis. So what happens is can lead to death of that corner as nothing reaches there, right? So why is that? It is because capillaries will have blood and blood reflects light back. It stops the way of light. So if your eyes had anything in front of it, then you won't be able to see, which is why the human body has adapted by not having any capillaries, because whenever light, if you shine light through your for instance, if you close your eye or try doing this on Abdullah, close his eye, shine light on it, it appears red, and you can sometimes appreciate some vessels as well, which means that if it appears red, it means there's some kind of blood or tissue fluid back there that is blocking the light and causing the red color, red shadow. It's basically absorbing light, which is what is proven as well, that this is why we don't have capillaries having anything in it, because we don't want the light to be blocked in any way. Otherwise you will see people, and the images of your veins and capillaries will also be seen. You will reflect them back as well. You'll see those overlapping over people. So do you get what I mean?
Yes, teacher.
Okay. So these are essentially the capillaries through the electron microscope. Just like we talked about a black and white detailed image. So you can see one cell thick, you can appreciate the nucleus. It is the relatively darkly scaling portion. The rest are the fibers and the tissue surrounding it. And yeah, that's basically it. So it's a little dull, which is why I can't show you the mitochondrion, but he has labeled that as well. And you can see how small it is. And he shows that there's one cell traveling through it at the moment as well. Okay, now how do veins and venules work? This is also a very important topic that you need to understand. Give me 1 minute. Okay, so veins and venules, like I told you, have very less walls, right? They need something to make sure that the flow of blood is maintained. There are two things that they use for this purpose. One of the things is valves. They have valves that are half crescent or semi-lunar shaped, which means it isn't a circular wall. It has half a circle and half a circle in both directions, right? These walls make sure that there is no backflow of blood. Blood does only flows in one direction, because like I told you, pressure in the veins is very low, but the volume is very high. So you need to, pressure maintains distance. Pressure, remember, pressure gradients cause diffusion, pressure gradients cause movement, pressure gradients cause mass flow. So if there is no pressure here, then what we need, systems to make sure blood does not go back, and deoxygenated blood does not start going back into the organ and pushing oxygenated blood away. How do we ensure that? Valves. And the second thing, the second thing is these muscles, our skeletal muscles. These, what they, not the muscles in the artery walls, these are the muscles on the bones and the arms, on the legs. These muscles, what they do is, they, as they contract, they come closer together and they become thick in nature. Contraction always thickens muscles. As they contract, the veins are naturally placed very close together to these muscles, because both our body and nature has this way of making sure that blood does not go back. As the muscles contract, they push on the vein and make sure that blood, there's a pressure exerted on blood to make it flow forward or push it forward, right? So that's basically it. And this can be understood by this question. So this question says normal venous pressure in the feet is about 25. When a soldier stands motionless at attention, the blood pressure in his feet rises very quickly to about 90 millimeter. So how does this pressure rise suddenly? And the answer is very easy. The muscles in our leg push against, whenever the soldier stands, muscles must contract, and to make sure that he's standing, and push against the veins to increase this pressure. Okay, so very easy concept. I have to take a meeting at 9, so this will be it for today. This was essentially our last slide as well. So we will do this slide tomorrow, but we are done with the first topic on vessels, the veins, venules, and the generic structural integrity of today's topic, okay?